We're going to start with the pitches focused on the topic on next-generation imaging and image-guided diagnosis and therapy for cancer. And the first presenter on my list is Antonio Scilimati. So if you'd like to go and you can speak from the podium there.
Thank you, Chairman. My name is Antonio Scilimati. I'm from Italy. I'm a professor of medicinal chemistry at the University of Bari. And as you saw, I'm here, and the call I'm referring to is next generation imaging and image-guided diagnosis and therapy for cancer. I am here to find partners and eventually API. To build a proposal to apply for ovarian cancer, an unmet clinical need. A few words on ovarian cancer. It is the 70th leading cause of death among women with ovarian cancer. There is no early detection, assay. Surgery and drug treatment are inconclusive. Many papers, many articles have been published, but there has been no real breakthrough in the treatment of women with ovarian cancer. Over the past 50 years, the overall survival rate has remained stagnant at about 30% at 5 years after the diagnosis. And if we compare with the breast cancer, the overall survival rate is 87%, so a quite different value. This very low survival rate depends on diagnosis made at an advanced stage, drug resistance, and incomplete surgery. It is therefore necessary to discover new drug targets, identify new biomarkers, and develop new probes for image-guided surgery. Then, if you are interested in this topic, please contact me and we will work together. Thank you very much.
Thank you very much. And the next speaker on my list is Linda Chaabane from Euro-BioImaging.
Thank you. So, Euro-BioImaging is a European research infrastructure that supports any scientist to develop his own research project by providing the access to imaging technologies across different facilities distributed in different European countries. We can connect you with different experts and their advanced imaging tools that can provide different imaging data kind applied to biomedical and medical fields. And we can also have these different scales starting from super resolution microscopy to microscopic resolution, and including also in vivo functional imaging at preclinical and clinical level. So we can also provide you some advice for your project and guide you through the different imaging technologies to select the most suitable one, and select the expert that can help you also to develop your projects. And we have this network of 150 imaging facilities that can provide different kind of services, starting from study designs, patient selections also, and animal models, and also including customizing new imaging probes or nanosystems for theranostics, which could be really useful for image-guided diagnosis and therapy for different purposes. We have experts of imaging in different fields, from cancer to cardiovascular and also neurological disorders. So we can really cover all the application actually. We would like to invite you to see, of course, our website, but also our marketplace where we put in place a different proposal actually from our network that will be pleased to be in contact with you. And of course, we are here for facilitating all the contacts. Thank you for your attention.
Thank you very much. Our next speaker is Iordanis Koutsopoulos from Athens University of Economics and Business.
Hi, everybody. My name is Iordanis Koutsopoulos. I come from the Athens University of Economics and Business, and I'm here today to talk to you about three novel uses of AI, which can be of use to any of the calls, either the two cancer-related calls or the cardiovascular disease one. So the current AI models we know operate under the assumption of having a massive data set. Now, what happens if this data set cannot be collected? So our team has expertise in a number of novel aspects of AI, and I would like to point to three of them. The first is online learning from data. So learning from data as they arrive in an uncertain fashion, in a streaming fashion, employing mathematical models such as multi-arm bandits, online convex optimization, and so on. So online learning is the first aspect. The second aspect is continual learning. So if you train a neural network to recognize, differentiate between cats and dogs, and then train it to differentiate between, let's say, foxes and wolves, and then you ask it again to differentiate between cats and dogs, it will have forget it. So it will forget it. This is catastrophic forgetting, and this is very important. This is an unsolved challenge. And so the thing is how to make AI models continually learning. And last, the third item is what has been mentioned also in a few other pitches, distributed learning and the capability to learn from distributed sources of data, geographically separated. So online, continual, and distributed. So our team focuses on these three aspects. It does real implementation of AI algorithms on devices and has also a number of expertise, other expertise such as building and implementing mobile apps, building AI algorithms and machine learning models on devices. Thank you very much.
Thank you. Okay, the next person on our list, we are not entirely sure if they are at the event, Stephen Gilbert. You're there? Oh, wonderful.
Hello, I'm Stephen Gilbert, and the title, I'm looking to coordinate a project, and it's got a preliminary title, which is Target Next Generation Imaging and Image-Based Guidance for Therapy of Cancer. And we look to build and coordinate a project around building an image-based automated quality pipeline using multiple modalities for the diagnosis and treatment of rectal carcinoma as a model cancer type. And we look to use histology, diagnosis and staging approaches from histopathology, machine learning, 4D CT planning, radiotherapy planning using online magnetic resonance, LINAC, linear accelerator approaches. These are all technologies that to some extent exist within our institute, which is Technische Universität Dresden, but of course we wish to bring in consortium partners, and of course a major challenge is getting the 45% funding. so major industrial partners, some of whom will we've already contacted. So I see this event a little bit like speed dating. I'd say I'm 50% likely to coordinate application. If we don't, and we don't secure those large industrial partners to get the 45% funding, of course we're interested in joining other similar projects which would make use of some of those technologies. What we bring to a consortium, so Professor Jakob Kater, new professor at the Institute, who has world-leading approaches in diagnosis from histopathology, staging of tumours, and swarm and federated learning within histopathology diagnosis. What I bring, I'm a new professor in the Institute, professor of medical device regulatory science. We see regulatory science as core to our project. We're looking to bring something which is close to the market approval and implementation through detailed quality development of pipelines. We're linking together existing technologies to a large extent. We are looking to introduce and explore new regulatory approaches. The last speaker mentioned online learning, adaptive learning. That may form part of this. This is a huge regulatory challenge, and anybody developing a project in this area needs to be looking at the regulatory requirements for such approaches. The other thing we will bring to this project is already approaches on online adaptive therapy on the magnetic resonance linear accelerator with centres in Dresden. So what's new in this project? I have 23 seconds left. Joined up workflow thinking and the close integration of quality, regulatory and patient care. What are we looking to add to this? Potentially partners who are very much involved in workflow thinking, PACS, partners, automated machine learning, sometimes approaches to workflow thinking, and partners in online tumour classification, particularly by magnetic resonance.
Great, thank you very much. And the next speaker is Rick van Heijningen from the VUB.
Good afternoon. My name is Rick van Heijningen and I'm representing the in vivo cellular and medical imaging group from the Vrije Universiteit here in Brussels. And that department is headed by Tony Lahoud. We have the ambition to coordinate a consortium and are looking for partners. We also could join another consortium. We have identified an innovative and simple clinical workflow for the image-driven diagnosis and stratification, optimization and monitoring of cancer treatment. This workflow and the components can be extended to the follow-up of patients and can facilitate image-driven personalized cancer therapy. The Vrije Universiteit Brussels is world famous for the invention and the development of single-domain antibodies, also called nanobodies. We would like to capitalize on this pioneering work and translate it in the field of PET and optical molecular imaging. In short, our goal is to develop contrast agents suitable for image-guided personalized cancer therapy and do so in a patient-friendly and cost-effective manner. We will show that nanobody-based targeted contrast agents in PET imaging can provide molecular information for effective therapeutic decision-making to tailor cancer therapy. We also believe that the same tracer with a fluorescent contrast agents can facilitate image-guided surgery and assessments. Optical probes will directly strengthen the visual aids for surgeons for better tumor resection, contribute to the detection and removal of metastases and lymph nodes, and to the overall stratification of patients. The tracers will be validated preclinically, and suitable tracers will be brought forward in early-stage clinical trials. We have gathered an international consortium which is complete for one specific cancer type. We would like to complement this consortium with AI-driven experts for the focused decision support systems that rely on imaging data. Further, we would like to reach out to biologists and medical doctors with a specific cancer-driven clinical need for which they want to develop PET and fluorescent nanobody-based tracers. We will also welcome approaches that directly integrate cancer imaging with therapy. Together, we will facilitate better image-based decision-making before, during, and after the treatment of cancer. The results of this project will directly benefit better personalized medicine and effective clinical decision-taking. It will also influence disease outcome, cost of treatment for healthcare providers, and quality of life for patients. Thank you very much for your attention.
Thank you very much. And our next speaker is Nuhan Purali.
Good afternoon, everybody. I'm Professor Nuhan Purali, representing Hacettepe University, located in Ankara, Turkey. Hacettepe University is the most established medical institution in the region. We are conducting education, medical research, health service, and technology development. We have 3 large-scale university hospitals conducting medical service and research at an international level. We have adult hospital, pediatric hospital, and oncology hospital. University hospitals has been accredited by the Joint Commission International since 2007, and they are regularly audited by the same commission. The annual numbers of the hospitals are huge. Yearly outpatient number exceeds 1.2 million, and inpatient number is above 60,000 per year. And we conduct over 70,000 surgical procedures. We have highly experienced health professionals, state-of-the-art medical equipment, and properly stored huge amount of medical data and sample collection. Those are ready for medical research. Further, we have a technopolis to translate the medical research into medical technology. We can take part in any one of the single-stage calls. However, we have already developed a small-sized consortium to apply for the second call, next-generation imaging and image-guided diagnosis and therapy for cancer. The use case we will use in that project is the human hepatocellular carcinoma. The project is expected to optimize available diagnosis and treatment systems, design, produce, and test new alternatives, improve patient outcomes, and reduce the human errors. The main innovations will be advanced visualization of the medical imaging data, federated learning for GDPR-compliant AI, multimodal data analysis, advanced self-educated education platforms for health professionals. And we are looking for a coordinator, a highly experienced coordinator. Also, we can take part, we can accept AI developers, healthcare providers, medical robotics developers, universities, research institutes, SMEs, and large industries, also pharmaceutical partners to enlarge our consortium. Thank you for your time and consideration.
Thank you. And the next speaker on my list is Cristina Dalmeida from MEDEXPRIM. We're actually going ahead of time, so 10 points to everyone for respecting the 3-minute limit. Thank you.
Good afternoon, everyone. I'm Cristina Dalmeida, Scientific Director at MEDEXPRIM. Today, I would like to talk about multidisciplinary tumor boards. If someone close to you have been diagnosed with cancer, you surely know how complex it is. Oncologists, pathologists, radiologists get together to analyze all possible treatment alternatives and to identify the most suitable one. In short, it's a forum where personalized medicine takes place. Tumor board meetings must be exhaustive. How could one evaluate all treatment possibilities otherwise? Now imagine how challenging it must be to make sure that all relevant patient information is gathered and updated, including clinical data, histological results, cancer cell mutation, genomics. Also, real-world data is most often unstructured and heterogeneous, and they can only be visualized by means of multiple tools that are connected to different data sources in the hospital. As a result, doctors spend considerable time gathering information and struggle to obtain the most updated recent data. At MEDEXPRIM, We want to solve this problem by means of a smart tumor board, allowing the simultaneous visualization of all data necessary in decision-making. Also, an onboard 3D organ model will be developed to help doctors to better access each case. We provide regulatory-grade and multimodal data sets by means of our software, which extracts, curates, harmonizes, and enriches real-world data generated in the hospital in compliance with GDPR. We will also constitute a dynamic data lake that will continuously feed the tumor board based on our expertise in radiology and digital pathology. Our research project will focus on hepatocellular carcinomas, which represent 5% of new cancer cases worldwide. This is a very aggressive liver cancer, which diagnostic is often belated. As a result, resection is the most preferred therapeutic approach. Our ambition is to promote a decision aid tool that can be deployed at all stages of the care pathway, from early diagnostics to therapeutic monitoring, in order to promote better care for patients. Building on our experience as a technical coordinator of the European 80-2020 project, Chameleon, we wish to lead a consortium for this groundbreaking project. Today, we are looking for partners holding expertise in genomics, data science, as well as a real-world evidence CRO to complete our consortium. Thank you so much for your attention.
Thank you very much. Next on my list is Blandine Hirtz, Gleamer. Anyone here? Ah, there you are. And just so the next people are aware, after Blandine, we have Ricky Sharma, Juan Verde, and Tamas Kitka. So if those people are in the room, you're up next.
Hello, good afternoon. So my name is Blandine Hirtz, and I'm in charge of grant and public affairs at Gleamer. So for this innovative health initiative program, we are interested in the next generation imaging and image-guided diagnosis and therapy for cancer call. So Gleamer is a French-based company founded in December 2017 by a radiologist, a serial entrepreneur in healthcare, and an AI engineer. And our role is to companion imaging users with AI-enabled workflow. Our solutions aim at detecting critical findings and alleviating the most tedious task to increase diagnostic performance and reduce interpretation time. So our solutions are fully integrated in the radiologist workflow and act as instantaneous concurrent reader to support radiologists and clinicians and direct patients in the right care pathway. So we have two initial solutions, BoneView and ChestView, which are deployed across more than 15 countries and 300 institutions, mostly in Europe and in the US. And so they are detecting major findings in trauma and chest X-rays, and they are granted by high-impact clinical validation studies. 2 of them are published in radiology. So we focus on high-volume application performed on X-ray and CT for acute and chronic conditions to detect, measure, and critical findings. So in this call, we would like to join a consortium, and what we can bring to this consortium is our strong knowledge in the radiology workflow, and mainly for specifically here for the chest, and with our solution, so ChestView, which is able to detect lung cancer and specifically nodule and mediastinal mass. So thank you very much, and I will be very happy to talk to you after. Thank you.
Okay, thank you, Blandine. And now I would like to invite Ricky Sharma from Varian to come to the stage, please.
Good afternoon. My name is Ricky Sharma. I'm a Vice President of Clinical Affairs at Varian and an Honorary Clinical Professor at University College London. I'm an oncologist. It's actually on this exact day 5 years ago that many of you would have tuned into the news and saw a burning high-rise building called Grenfell Tower. And there were 72 unnecessary and preventable deaths that morning in London. And if you think about lung cancer, there are 95 deaths every single day in the UK from lung cancer. And some of those deaths are preventable. If we were integrating information that we actually have in the clinic, but we don't have a means of integrating it, we could be preventing some of those deaths. So this proposal is about bringing together all of that information into a cloud-based platform. So that's, for example, radiomics, genomics, other omics, into a cloud-based platform which Varian and Siemens Healthineers will provide. And then using those data to derive algorithms to improve our decision support tools. So in order to deliver on that, we are looking for partners, both academic organizations with clinical data sets and industry partners in terms of the radiomics, the genomics, and other omics platforms. And ultimately, we would like to include patient groups in terms of patient-reported outcomes and patient advocacy, and also small and medium enterprises in order to deliver on some of those newer technologies. So I hope you'll join us in terms of personalizing non-small cell lung cancer and making the clinical outcomes significantly better. Thank you for listening.
Juan Verde. No worries.
So, hello, my name is Juan Manuel Verde. Thanks a lot for this opportunity. Sorry for the delay. I'm a surgeon coming from Strasbourg, from the Institute of Image-Guided Surgery at Strasbourg, which is an institute fully conceived for the translation to cancer patients of image-guided therapies. In this project, we are aiming to deliver a solution with a low cost footprint, which should be interoperable and relying on AI to address the full spectrum, starting from planning, the full spectrum of the patient journey, starting from planning, including decision-making, moving all the way into the intraoperative conditions or intratherapy conditions, and also aid with prediction of outcomes, with the prediction of outcomes. For this, we chose ultrasound because of its many advantages, including its real-timeness, including its multimodality, meaning that we can assess ultrasound or we can acquire ultrasound images by the means of a sensor in the tip of an endoscope, a laparoscope, or a handheld transducer. But it's also multi-scale, meaning that we can assess an organ, a tissue, all the way down to the cellular level. But even more important is that ultrasound is widely available, maybe due to its low cost, but it's also almost universal. Across different modalities, across different geographical conditions, ultrasound is present always at the edge of the point of care. We also know, and we learn by experience, that ultrasound is very complex. The analysis of images is very complex, and the learning curves, due to its interoperability, it's very large, or they are long, sometimes requiring years, if not decades, to become proficient. And this is why in the Institute we embrace AI, just trying to democratize knowledge and democratize as well experience. And we already established some proof of concepts in the field of endoscopic ultrasound navigation, in the field of automatic image recognition, as well as in the field of needle guidance. We already have an entire team of around 40 computer scientists working on AI for ultrasound on a daily basis. We have full translational capabilities starting from conception all the way down to clinical translation. We have 2 platforms, preclinical and clinical, fully equipped with advanced medical imaging. And we also have the experience required to coordinate a consortium. And this is why we are proposing the IHI to coordinate a consortium trying to address the call and trying to use AI and ultrasound to cover the full spectrum and to make ultrasound interoperable in the field of cancer imaging. Thanks a lot. 3 minutes. That's it.
Okay, thank you very much. Tamas Kitka. Is Tamas in the room? Yes, excellent. So I should just add, it's already up there on the slide, but you can see this is a pitch relating to the other cancer call that we have. Tamas, go ahead.
Hello everyone, my name is Tamas and I'm here to introduce the Vascular Research Group's proprietary chlorotoxin analog program to you. So as you may know, the metastases are usually detected using imaging solutions, for example PET-CT, but the tracers usually used do not fit the needs. For example, the most frequently used one, the FDG, marks tissues with high metabolic activity, but if the metastasis is surrounded with tissues with similarly high metabolic activity, it cannot be found, and new treatment won't be initiated, and of course it can lead to fatal consequences. So what we started with is to use MMP2, matrix methylprotease 2, as a marker for tumor tissues, especially for metastasizing tumor tissues. Generally, the matrix methylproteases are expressed ubiquitously, but this MMP2 is very specific for metastasizing tissues. And also it has a reuptake mechanism that allows our compounds to be accumulated intracellularly. So that's how we can mark the tumor cells. We started from chlorotoxin, which is a peptide toxin. So it is under clinical development in 2 projects. So they think it would be working for a CAR T-cell therapy and also as a fluorescent paint. But it's good to be noted that the chlorotoxin has quite moderate affinity and also moderate selectivity towards MMP2 that, as we see it, increases serious questions about its clinical applicability. But fortunately, we succeeded to increase both the affinity and the selectivity, and with this we reached 3-4 times increased tumor uptake versus the original chlorotoxin, and we also increased good selectivity for tumor cells versus non-tumor cells. We also had an in vivo proof of concept study with a fluorescent paint in a mouse model. Now we are also starting creating new chelator conjugates for radiolabeling, and that's where we need partners because we are now entering a new field. We have some cooperations, but we are looking for new partners. We have the target molecule, a targeted molecule, and we have the whole idea. We need to find other companies to walk around the route. Thank you very much.